Hello, good afternoon. Uh, Happy Women's Day. This is the voice of Olatunde Michael at Om3ga Bio-Edutainment. This afternoon, once again, we have come to our classroom settings, and that is going to be microbiology essentials, the foundation of the invisible world. In this special edition of our lecture series, we are going to touch the following. This is going to be a recap of the whole work we have done, the whole topics we have actually taken care of in introduction to microbiology. Don't forget, this is Om3ga Bio-Edutainment Hub educational series. At the end of this revision session, students as well as viewers at home should be able to define microbiology and explain its significance in science and the relevance of microbiology in daily life. After that, the student is expected to identify major groups of microorganisms, which include bacteria, fungi, viruses, protozoa, and algae. After that, each student should be able to outline the historical milestones in microbiology, that is, history of microbiology, with a very special focus on key discoveries and their impacts in our daily life. Another objective of this revision session is that students should be able to describe the scope of microbiology, which includes its application in medicine, industries, and the environment, including how microbiology can be applied in our everyday life. Then, after that, students should be able to explain basic microbiological techniques such as aseptic handling, staining, and microscopy. Thereafter, the students should be able to differentiate between prokaryotic and eukaryotic microorganisms with So once again, I want to welcome you to special edition of our revisional exercise of Introduction to Microbiology. Don't forget that the first set was aired behind the camera so that we will be able to actually see one or two other important focus that we need to do. Don't forget that today, just like I said, this lecture is actually titled Microbiology Essential, which is a foundation of the invisible world. In this revision exercise, we are going to achieve at least 6 important things. The first one is we should be able to define and explain microbiology, the significance in science and daily life, and so on and so forth. So without wasting our time, we should go straight to the educational series. So which has, this is general microbiology. So the first thing is, don't forget that this one is exam focused. That means we will be able to give you some shortcuts to understanding certain things in microbiology. So the first, I'm going to give you like 3 important things. The first one is 3 important sentences that can help you to understand some certain things in microbiology. So viruses play around. Then the second sentence is sanitize. Sanitize before touching everything. Third one is the third one is CIVS. So you will pick this one from the word civilization. So when I come back, I'm going to give you so I want you to take note of these three sentences that will really help you to be able to master a lot of things in the general microbiology. So the first one, which has to do with a big is standing for bacteria. F is standing for fungi. V is standing for viruses, and P is standing for protozoa. And A is standing for algae. So this sentence will help you to be able to remember classification of microorganisms. So they can be classified into bacteria, fungi, viruses, protozoa, and algae. All these are microscopic. That is, you cannot see them without the help of your naked, without the help of microscope or lenses. So example include bacteria, E. coli. To be able to give example in each case, yeast. This one is HIV. This one is Plasmodium, malaria, Plasmodium, and algae such as Chlamydomonas. So that is that. So the second sentence that we have here is S is standing for sterilization. S is standing for sterilization. B is standing for biosafety. That's containment, biosafety. T is standing for transfer techniques. Transfer techniques. Okay. While E is standing for environmental. So this is actually talking, if you do not understand the meaning of what we call aseptic techniques. Aseptic techniques can be gotten from this forward sentence. S, that is sanitize before touching everything. You should know that S is standing for sterilization. B is standing for biosafety. T is standing for transfer techniques. That is transfer techniques. Then E is standing for environmental. Okay, environmental control. Okay, you should be able to know that. Then the last one, which is CIVS. So this is actually talking about gram staining techniques. If you are asked to gram staining techniques or gram staining steps. So the first one, C, is standing for crystal violet. Okay, crystal violet, which is our primary stain. This is going to be the primary stain. You must have heard me talking about primary stain, secondary stain, and okay. I is standing for iodine, which is serving the purpose of mordants. Okay. Then V is standing for alcohol, which is going to serve as the colorizer. Then S, this S is standing for safranin, which is going to be the secondary, or safranin. Okay, which is going to serve as counter stain. So what this means is that the first thing you will add is the primary stain. We had it in such a way that by the time you add it to your specimen, whenever you add it to your specimen, all of them will take up the crystal violet. That is the primary stain. So the color of the crystal violet is going to be stained. You are going to notice it here. So immediately after that, you now add a mordant, which is going to be an iodine. What does mordant do? What they normally do is to make sure that they help the primary stain to stay fast, to adhere, to be able to stay adder. So that is a mordant. So immediately, if you now add alcohol, which is going to decolorize, so the alcohol is going to serve the purpose of removing all these stain, that is the color of crystal violet as well as the color of iodine. So if the color remains, okay, so after that, that is the colorizer is going to remove that. So when you now add safranin, if this color has been removed by iodine, so that means the final color of your sample is going to be safranin. But if your sample has actually taken the color of crystal violet, so even if you had alcohol, the color, the primary color will still stay there. So if it stay there, the color of safranin is not going to be to have any effect on it. So the color of the primary state will remain, and that is for gram positive. So the color, the primary color will remain even after being decolorized and after counter staining. But if it is now gram negative, if it is gram negative, that means the sample is going to take up the color of safranin. That's why they call it what? That's why they call it gram negative. Now, don't forget the difference between gram positive and gram negative. I told you that this is from A to B. A to B is the cell wall or cell membrane of bacteria cell wall. Now, in between this one, the outer cell wall and the inner, is a compound that they call it peptidoglycan. Okay, so the thickness of peptidoglycan on the cell wall will now depend on whether it is going to be gram positive or it is going to be gram negative. Don't forget what I said. Peptido from peptide, that is coming from protein, and glycan, that is a glucose. That means the combination of a protein and carbohydrate on the surface between the outer cell wall and the inner cell wall is called peptidoglycan. So if the layer of peptidoglycan is very, very thin, if it is very, very thin, so the primary gram stain, which is crystal violet, if it is too thin, that means it will be very easy for alcohol to remove because it is very, very thin. So it is going to be so even when you had primary stain, if it is too thin, it is going to be gram negative at the end of the day because of the very thin peptidoglycan layer. But if it is very, very thick, that means it is going to be gram positive, thickest peptidoglycan. So that is the major difference between gram negative and gram positive. I've told you severally that you should know. One thing you should know is that you should be able to know CIVS, C crystal violet, which have as the primary stain, and I, which is iodine, which is going to be like a mordant, and V is going to be alcohol, which is going to serve as a decolorizer. So then the last one is a safranin, which is going to be a counter stain. And I said that the difference between what make gram positive to be different from gram negative is the thickness of their peptidoglycan layer. If the primary stain, which is the crystal violet, if the color is retained at the end of the staining technique, gram staining procedure, that means it is going to be gram positive. But if the color is going to be safranin, the color of safranin, that means you should be able to get the exact color of your dyes. So the primary stain and the safranin is very, very important. So you should be able to know that. Okay, so that is that about some of the mnemonics. That is how you can remember at least 3 things that are very, very important in microbiology. So which is the first one is big friendly viruses play around. B is standing for bacteria, F is standing for fungi, viruses play, that is protozoa, and around, that is for algae. So then another one for you to be able to remember what aseptic techniques also entails is you should be able to remember sanitize before touching everything. S is standing for sterilization. You have to sterilize, you clean, you remove. Don't forget that aseptic technique is how can you work and make sure that you eliminate, you eradicate contamination. That is the basis or the basic of an aseptic condition. So once you remember sanitize before touching everything, that one is going to give you what aseptic condition always entails. Okay, now thereafter don't forget that I told you the difference between gram positive and gram negative is that the color. Okay, what color? The next question, if I were you, the color, what is the color of gram negative? The color is going to be pink. It's going to be pink. And the color of that is gram negative. Gram negative is going to be pink. Whereas gram positive is going to be purple. Gram positive is going to be purple. So you should know this at this level, peptidoglycan layer, which is an outer layer. So depending on the thickness of the peptidoglycan layer, so this is outer cell wall. This one is inner. So between here and here is called between here and here is called okay. So between here and here, that is the peptidoglycan layer. So the thickness will depend on whether it's going to be gram positive or it's going to be gram negative. If it is the layer, peptidoglycan layer is thick, that means it is gram positive. If it is thin, that is gram negative. Okay, so I want you to really take note of this. So the next thing is how I'm going to give you like 3 or 4 questions, and what I'm going to do is that that you can add under this. Okay, I'm going to dictate it. Okay, I'm going to dictate it, and let's see what is going to happen. Okay. So I want you to really take your time and take this by your. So you don't need to cram too many things. You don't need to cram without stress. So I'm going to really give you 4 questions, and I will ask you question 1. Okay, why do we gram stain? That is, what is the essence of gram staining techniques? Why do we gram stain in microbiology? That is the use of gram staining. So don't forget that I have just told you that the reason is not far-fetched. The reason is that gram positive, it just because of the thickness of their cell wall. If the thickness of the peptidoglycan, if it is too thick, that is gram positive, and if it is too thin, that is a gram negative. And I will love you when you want to answer this question. You should be able to know what do we understand by gram staining. What is the basic principle? So when you want to answer this, I'm expecting you to know the definition of gram staining. Okay. After that, basic principles. Basic principles of gram staining. You should be able to highlight it. Then you should be able to differentiate between positive and negative gram staining. How would you see that under microscope? It's very, very important. So another possible question is that can you explain the difference between sterilization and disinfection? So this is a type of aseptic condition. You should be able to know the difference between sterilization and disinfection. So from this word, sterilize, that is, you remove. It has been contaminated. It is expected to have been contaminated. That's sterilization. It is expected to have been contaminated, but you will remove contaminants. So it's a process whereby you apply sterilization techniques, or you apply some techniques in order to remove contaminants from surfaces. Okay, so that is that. Then disinfection, that is, you prevent. This one is a removal, this one is a prevention. You prevent. So if you now want to so this one is talking about kill, outright kill. This one is talking about destroy. You destroy, you eradicate. This one is destroy. Sterilization is to destroy. Disinfection is to kill. Okay, it's to kill. So you should know that. Okay, that's where you apply the second thing I told you. Okay, that you have to remove, you sanitize before touching everything. That is the level you will apply that. So this another question now is to look at what are the importance of aseptic condition. How important is aseptic techniques in microbiology? How important it is? Okay, so the first thing is don't forget, you have to tell us, define aseptic condition or aseptic technique. You should be able to define that. Then you should be able to tell us the importance before you move to the importance. And the importance is that it helps us to it protects the surfaces. Protect, apart from the samples, to protect the laboratory surfaces, our microbial culture, that is samples, as well, and the personnel, that is, and you self, personnel. So aseptic condition help to protect our surfaces, the laboratory surfaces, the culture, that is the sample, and you as a laboratory personnel. It's going to prevent you or protect you from getting contaminated or getting infected. Okay, so another question is to mention. You have to mention 2 staining techniques. If you have to mention 2 staining techniques and their importance. So at this point, I'm very sure that the first thing you will do is to mention gram staining, which stain the cell wall. That one targets the cell wall. Another kind of types of stains or staining technique that is not mentioned is endospore. Endospore. Okay, the one that we only target the microorganism that form endospore. Okay, so example is the one they use for Bacillus species, because Bacillus is known to form endospore. They will form a kind of a protective coat around them. So you target endospore. Okay, those that form endospore. Then another one is acid fast. Acid fast stain. So that's the one they normally use for Mycobacterium tuberculosis. Mycobacterium tuberculosis, because Mycobacterium tuberculosis, they are, apart from having very thick peptidoglycan, they also have other things. They also have other layers, like they have other layers that will give them additional protection. So making them, so what they normally do, the basic principle in acid fast is that the first thing is they will use acid to destroy this before they will now apply gram staining techniques. Although just know it at this point, you don't need too much to know too much on that, but those are the 3 types of stains. You just pick any 2 from there. If you are given a question to mention 2 staining techniques and their importance or their basic principles, I've told you the first one is gram staining techniques, and that is the one they normally use to differentiate into whether they are gram positive or they are gram negative. And the second one is endospore staining, the one that they use for Bacillus and some other types of bacteria like Clostridium. Okay. Then the third one is acid fast, the one they normally use for Mycobacterium tuberculosis, because of the additional protective layer that you find in them. So that means they will first of all use acid to dislodge or break down that kind of a layer before they will now apply the other one. So example of that is acid fast. Okay, so now I want to now go into some key topics, apart from other key topics that we have in microbiology, or which we have touched all together in this. So the first one, I want to divide it into 3. I want to divide it into 3 basic topics. The first one is key figures in microbiology. That is, what are the very important microbiologists that you need to know when we are talking of the scope, the history of microbiology. So I'm going to give you the shortcuts to be able to remember that. Okay, then followed by the scope of microbiology. So I'm going to give you also mnemonics to be able to also understand and remember that. So followed by types of culture media. So after that, I'm now going to give you some likely questions, very likely questions that you need to go and read more about them. Okay, for this examination, especially exam that involve general microbiology. Okay, now let me just allow for just 2 minutes break on this, okay, before I come back, so that you'll be able to write what you have on the board. Take note the board. So I want to welcome you back to the continuation of this. So if you are given a question like you have to discuss the historical milestones in microbiology and what are their contributions in modern day science. So you just want to, what you should do is just to remember LPPK. Once you remember LPPK, L is standing for Leeuwenhoek, which is the father or the foundation. No, the father. I told you during the normal class that Anton van Leeuwenhoek, who happened to be, is the father, the person that discovered microscope, and I gave you some of what he said about that. Okay, so the second P is Pasteur, Louis Pasteur. So Louis Pasteur is very, very notable because of three important contribution of his into the world of microbiology, and which is still very relevant in microbiology till today. The first one is germ theory. Louis Pasteur was the one that actually came up and said that diseases or infection is caused by germ, something that is too small to be seen by our naked eyes. Then apart from that, I told you about the controversy that surrounded the spontaneous generation, and which I actually expected you to still go more and read. Then Pasteur is still very very relevant and very popular because of the theory of pasteurization. So read that. And Petri, that is the discoverer, somebody that could discover Petri dish. So that's why when you want to culture in the laboratory, you prepare your agar inside Petri dish. Now we have disposable, the one that you just use and discard. Okay. Then the last K, which is a Koch postulate. That is a Koch postulate. You should also try and remember that. Okay, that's very, very important. Now another thing is another likely question is if you are given another question like this. If you have a question that to describe types of culture media and their uses. Okay. The first one, so you should be able to mention liquid media, and liquid, I told you, means broth. So, and that one can be used to grow bacteria from liquid samples like the blood. Okay. Then the second one is solid. I made mention of solidifying agents. That is agar. So agar, which is used for isolation. You can use it to isolate, as well as you can use it for identification. On it, if microorganism starts to grow on it, you could actually use it to be able to identify to certain level. Okay. Then number 3, I made mention of a general purpose agar. General purpose. I made mention general purpose is for general. I told you that for general to isolate both bacteria and the fungi and other things will grow on that. And I give you example of that. That is, if what you just want to know is just to you are not interested in identification, you make use of general purpose agar like nutrient agar or nutrient broth. Okay. Now this another one is selective. From the word selective, you know what it means. That is, you select against certain microorganism. So you prevent the growth of certain microorganism, when you allow the growth of that. So I gave you an example, like addition of certain dyes or antibiotic to prevent the growth of gram-positive, whereas gram-negative will grow there. So there are other agents too that you can also use to prevent the growth of gram-negative while you allow gram-positive to grow. Okay, I gave you an example of that. I made mention of McConkey. Okay, McConkey agar. I said something about SSA, Salmonella Shigella. I made mention of mannitol salt agar, MSA, MRSA, and so on. MRS, sorry. So and so on. Then another one is differential. I said McConkey is both selective as well as differential. Okay, differential in the sense that can differentiate between lactose fermenters as well as non-lactose fermenters. So then I made mention of enriched. That is, you enrich your general purpose agar with blood and serum and some other. You can also enrich it with eggs. Once you know that such microorganism would definitely prefer certain material, so you will just enrich your agar. And I gave you an example that it's just like when somebody is taking garri, and you know garri is pure starch or carbohydrate. So by the time you enrich it with milk, that's protein, with granules, that is another protein and fat. So that is enriched. So, and I gave you an example like a blood agar and chocolate agar. So I want you to really mention something about this. So this is what I am expecting you to say under, if you are given that to describe types of culture media and their uses. So I'm expecting you to give us the basic principle. You list them first, then you give examples. By the time you give example, now give us the function. That means if you don't want to use it, you can also tabulate name of agar or culture media, name of culture media, examples and their functions. So that is what I'm expecting you to do. Let me take like 1 minute, 2 minutes break. So once again, you are welcome back. Let's consider a situational case where we have another question. Another question that says, explain the roles of microbes or microorganisms. The roles of microbes in human diseases. So we are not to focus in animals, we have to focus in human disease. Okay. Now, basically, I want to divide microorganism into four. One, two, three, four. Okay. The first one is a pathogenic. Microorganism can be pathogenic. The second one is a opportunistic microbes. The third one is toxigenic. And the fourth one is a microbiomes. Once you remember POTM, that is pathogenic, opportunistic, toxigenic, and microbiomes. Okay. When we talk about pathogenic microorganisms, these are organisms that have the ability to cause, to initiate infection, to cause diseases. So they have the ability to initiate or cause infection. An example is a mycobacterium. Okay, Salmonella, etc. So they have that ability. So what they will always do in their life is to cause and initiate infection. So that is that of opportunistic. That is that of pathogenic. Now the second one is opportunistic, from the word opportunity. Initially, they don't have the power, they don't have it in mind of causing what? Of causing an infection. Example of this I mentioned that in the class during our physical contact time. I told us about what is called normal flora, the microorganisms that you have them present on the surface of our skin or on the surface of certain surfaces as normal. They go around their normal businesses without causing any harm. But in certain conditions, especially when their host immune system is very low, so they can now begin to cause infection. They are taking advantage, and that's why they call them opportunistic. And example of that includes Candida species. So this is a yeast. Under normal circumstances, this yeast Candida is a normal flora in the vagina. It's a normal flora in female. Okay, so they are there to, as a normal, they just go around their own without causing any infection, without causing any harm. But it is these Candida, once they have reached certain population, or the immunological state of the host is very, very low, or when the host refuses to fulfill what is called personal hygiene, So the immune status of the host can go down. Thereby, Candida will now begin to multiply. And by the time they multiply, they will now cause an infection. And that example of that is toilet diseases in female. So it's as a result of this. Okay. Now, toxigenic, from the word toxigenic, toxin, they secrete toxin. So example of that are food. So we have that of toxigenic bacteria in food poisoning. Example of that is Clostridium species. So they are found to cause food poisoning. Okay. From the word toxigenic, they produce toxin, they release toxin. I told you the other time that there are two basic types of toxin. Okay, we have endotoxin and then we have exotoxin. So example of toxigenic bacteria is a Clostridium species. Okay, now the last one, which are called microbiomes. Microbiomes are population of microorganisms that exist in close association with human, especially in the gut. They form is like microorganism in their community. They form a kind of community in the gut of a human. So thereby leading to diseases. So microbiomes. Example of that. So they cause a kind of gut imbalance. They exist normally in the gut, and one of the reasons why they exist in the gut is because they also help us. They perform certain function. They help us in they are beneficial in certain areas like the digestion. They help us in digestion of vitamins. They help us in the production of even fighting against some infection. So they exist normal in the gut of human, and whenever the population now reduces, their population, when it reduces, it will now form a kind of gut imbalance, and that kind of gut imbalance is a very it's a disease in human. So that is an example of microbiomes. An example of this is a Clostridoides species. Clostridoides species. Okay. So having said that, I'm expecting you to pay attention into what we call pathogenicity. I mentioned that pathogenicity, you should be able to talk about immunity. What do we mean by immunity? Immunity is inability to establish diseases or infection in a host, or prevention of establishment of an infection in a host. So that is immunity. Okay. Then apart from that, I want you to you should also mention what is called personal hygiene. How can you use personal hygiene in so you should be able to, apart from when you have to explain the roles of microbiomes in human diseases, I'm expecting you to divide them into four: pathogenic, opportunistic, toxigenic, and microbiomes. And you should be able to give examples in each case. Then apart from that, you also need to explain some basic terms and terminologies, as I also explained for you in the last video. Okay, so because of our time, I want to give you let me just mention. Okay, let me just write another question. You have to go and develop this on your own. List basic laboratory techniques that you know in microbiology. Okay, and what are their importance? You have to list and explain. List basic laboratory techniques in microbiology and what are their importance. Under this, I'm expecting you to mention aseptic techniques, talk about staining methods as well as microscopy. You should be able to talk about sterilization and disinfection. Then I'm expecting you to be able to talk about culturing techniques or cultivation of microorganisms in microbiology. So just like I said, and I gave you an example on the preservation techniques and their importance, I want you to, you have to submit the assignment, I think tomorrow. So having done that, I want to really believe that you will be able to, once you are able to do this, you'll be able to pass your question. I mean, you'll be able to pass your exam in microbiology. Pass your exam in microbiology. So if you have any question or query, I'm expecting you to really make comments in the comment section. Okay, so it is on that note that I want to really thank you for your listening attention, and I want to see you in our next video. I want to see you smiling in your examination. So once again, thank you, and God bless you.